What time do you make it, Frank? I mean, your time?
It's 2 minutes past.
It is. Okay. All right. Hello, everyone. My name is Dr. Henry Greathead, lecturer in animal production science at the University of Leeds. It is my great pleasure to welcome you all to the 8th virtual seminar of the Global Food and Environment Institute. And the seminar is entitled, Listen, We Can Reduce Heat Stress in Livestock Through Nutrition, which will be presented by Professor Frank Dunshea, Professor of Animal Growth and Development at the University of Leeds. The Global Food and Environment Institute, or GFEI, is an interdisciplinary research community which brings together members from across academia, industry, and public policy to work on the integrated challenges of food security and climate change. For further information, please visit the GFEI website, and you can find the URLs or links to the website in the chat bar. This seminar is part of a series of seminars, so please check the chat function with the link to our series advertised on Eventbrite, and the website and our Twitter feed for more information. And again, all those URLs are available on the chat in the chat feature of Zoom. Before I introduce Professor Frank Dunshea, I would like to remind you of the seminar etiquette. Please ensure that your cameras and microphones are switched off for the duration of the seminar. The presentation will take about 20 minutes, during which please use the chat function addressed to everyone with your questions. So at any point during Frank's presentation, please type in your questions. And then at the end of the presentation, there will be 10 minutes or however long we need for the question and answer session, during which I will collate the questions from the chat function. May I remind you that the seminar is being recorded, and a recording will be made available via YouTube. So now I introduce Professor Frank Dunshea, Professor of Animal Growth and Development at the University of Leeds. Frank joined the University of Leeds in August last year, so 2020. He spends 20% of his time with us, currently remotely, and his remaining time in Melbourne, Australia, where he is a Redmond Barry Professor and Chair of Agricultural Science at the University of Melbourne, which is where he joins us from now, where I believe it's 9 o'clock in the evening. So over to you, Frank.
Good. Thank you very much for the introduction, Henry, and it really is a great pleasure and privilege to be speaking to you today on a topic that I spent about the last decade working on heat stress. Obviously, in Australia, it is a big issue because of the hot environment that we face every summer. But it actually is becoming increasingly important all around the world, including the United Kingdom. So why are we interested in heat stress? Well, as we know, climate change and global warming has resulted in more heat events occurring, and they're actually occurring more frequently, and they have actually greater duration. So it's not only the average temperature that's increased, it's also the number of heat events. And in particular, it's when the nighttime temperature doesn't get down below, say, 23 degrees, where the animals or the humans can't dissipate heat, that we really run into problems. As I said, it's a big issue for us in Australia, but it actually is a big issue in the UK. So if we look at what happened last year, there were close to 2,500 people in the UK who died as a result of heat stress. This is particularly prevalent amongst people who have comorbidities. So people who are obese, people who suffer from diabetes, for example, are really susceptible to heat stress. So, I'm talking about animals here, but I think it is also critical for humans as we as a society, as we age, as we develop these a sedentary lifestyle, and we develop these diseases, it's going to be actually become a greater issue for the human population as well. So most of you will recognise this idyllic setting, the hay wain, a painting by John Constable. Whenever I'm in London, I go to the National Gallery and sit in front of it for a few minutes. But this is what it looks like nowadays. So this is the same setting, the River Stour. And what we see during hot days now is that farm animals seek out water. So here you see a dairy cow trying to cool itself down by standing in water. And we see this quite often on our dairy farms, and you'll see it in dairy farms throughout the UK when we have these hot days. Just before I go any further, though, I'd like to sort of, I might use these terms through my talk. I'll talk about the thermoneutral zone. So that's the range in temperature, humidity, and activity that an organism can withstand, where their body temperature, they can maintain their body temperature by losing heat to the environment through sweating, through panting, wallowing, or through evaporative loss through the skin. Heat stress conditions occur when the temperature and humidity or activity gets beyond that, where they can control their body temperature, results in an accumulation of heat and eventually heat stroke. And if you reach heat stroke, then the situation can be diabolical. And this is a good schematic I like to use when I'm describing what happens in farm animals when they're exposed to increasing levels of environmental heat. So as we move across here, where this arrow becomes more red, we're increasing temperature. What an animal will do to reduce metabolic heat is to, first of all, reduce their feed intake. Classic sign that we see during high temperatures. They'll reduce their activity, so they're not generating metabolic heat. And in lactating animals, they'll actually reduce the amount of milk that they produce. And all of these are directed at reducing metabolic heat. And some of the nutritional strategies that we've investigated have actually focused on how might we assist in reducing metabolic heat. Another thing that happens is that, in fact, blood flow goes away from the gastrointestinal tract and the internal organs to the periphery as the organism tries to dissipate heat through the skin. So by partitioning blood away from those internal organs towards the periphery, it can increase radiant heat loss and therefore cool the animal down. But a consequence of this reduced blood flow in the gastrointestinal tract is that, in fact, the gut becomes hypoxic. What that means is there's low levels of oxygen and we get oxidative damage. And very quickly we can get increases in gastrointestinal permeability and invasion by organisms or other molecules that may be existing in the gastrointestinal tract. So we've also looked at nutritional strategies to try to arrest this oxidative damage. The other way that many species dissipate heat is to increase respiration rate. So to increase respiration rate, and therefore they can dissipate heat through evaporative heat loss. And this can also result in some effects on the animal physiology as well, where we can actually get blood alkalosis, which can result in maintenance energy requirements going up. And so some of our other nutritional strategies will focus on this evaporative heat loss. Today, just because I've only got 20 minutes, I'll really focus mainly around this hypoxia and the use of antioxidants to improve the animal's condition there. So this is an example of what happens in the human and in the dog. So the intense red here is blood flow. So you can see that there's blood flow to the lungs and to the gastrointestinal tract during cool conditions. That's where a lot of the blood flow is partitioned towards, similarly in the dog. But when exposed to high temperatures, you can see there's much more blood flow indicated by the red colouring towards the periphery as we try to get rid of heat from the body. If you look at what happens in the dog actually tries to dissipate heat through panting. And, sometimes, a dog might be panting at something like 200 breaths per minute to try to dissipate heat. And I challenge anyone to breathe at 200 breaths per minute for any length of time before they become exhausted. And this is just another example here. So this is blood flow to the forearm of human subjects that are exposed to either thermoneutral conditions or they're heated. And you can see that oral temperature goes up, as does blood flow to the skin. It's maintained while the heating is maintained. And then once the individual is cooled down, blood flow goes away from the skin back towards the periphery and temperature drops, oral temperature drops. So, just another example graphically of what I'm talking about here. And if you look at, you can see this visibly here. Here's a pig that's maintained under thermoneutral conditions, mouth closed. If you look here, you can see that this pig exposed to a temperature of 35 degrees. The skin looks a lot pinker because the blood's being dissipated towards the periphery. Open mouth, panting, rapid respiration rate, probably up around 150 to 200 breaths per minute, and some salivation. And so I'm talking about a difference between 22 degrees and maybe 35 degrees. And this is quite clear indication that this animal is in some sort of distress. And these data were from Sarah Pierce from Iowa State University in the United States. I think it's quite a good example of what happens during only 6 hours of heat stress. So these cross sections of the small intestine at the ileum, if we look at 0 hours, so this is from a pig that's maintained at about 22 degrees Celsius, well within the thermoneutral zone. And this is what we would say would be a healthy small intestine. So these structures here are villi. You can see they're long and extended. And this is the site of absorption of nutrients. And this increased villi height, these high villi heights, means that there's a great surface area for nutrients to be digested. And there's also digestive enzymes that exist in proximity to these villi that also aid in digestion. And down here, you can see that between the villi, it's quite tight. There's no sort of opening up of any of these junctions here. So that is protecting from invasive molecules from the small intestine if they exist. But even after 2 hours of being exposed to a high temperature, you start to see the villi sloughing off. So they're losing their structure, and you can see clearly opening up of these tight junctions here. 4 hours, the villi are virtually non-existent anymore, and you can see by 6 hours these tight junctions are not tight anymore. And what happens here is you get invasion by endotoxins. Bacterial endotoxins can enter the bloodstream. Digestion ceases and or slows down considerably, and the animal will be suffering from some pyrogenic responses to the invasion by these endotoxins. So this is what happens in only 6 hours. So this pig was exposed 22 degrees, temperature was increased to I think 40 degrees, and 6 hours later, this is what we see. And a classic example of what we see in humans is runner's gut. So anyone who's looked at some of the Olympic events, the marathons, or the long distance walking events, you'll often see bloody diarrhea running in the legs of some of these athletes as they're suffering from something very similar. So I've used an animal example here, but this quite clearly happens to humans as well. So that is largely, those responses in the gut are largely a result of the failure of the antioxidant system. So we have a system of production of reactive oxygen being balanced with antioxidants. So it's normal for the body to produce these oxidants. It's when this balance gets out of whack that we run into problems. So oxidative stress really is the excessive production of reactive oxygen or a deficiency of antioxidants. And when we lose this balance, we have a failure of the antioxidant system, the animal, and we get resultant oxidative stress. Our initial work in this area focused on two antioxidants, and we chose these for particular reasons. We looked at vitamin E, and the reason we did that was because it's the major lipid-soluble chain-breaking antioxidant in cellular membranes. So it's actually contained in the membranes itself. And it directly scavenges superoxide and singlet oxygen. And the other compound we chose to look at was selenium. And we chose selenium because it is a component of many enzymes, in particular enzymes of the glutathione peroxidase system, but also other systems that are involved in protection against oxidative damage. So these are the two that we chose in our initial studies, and we've done quite a bit of work in this area. And I'll just show you a few slides to sort of give you an example of the sort of effects that we might see. This is a typical model that we might use. This is actually looking at heat stress in sheep. On this axis here, I have respiration rate, and then hour of the day on this bottom axis. And then we have exposure to high temperature. This time it was, I think, 36 degrees from about 9 o'clock in the morning until 5 o'clock in the afternoon. And then we reduce the temperature down to around 26 degrees. And we use this model to try to simulate a heat wave. And the various treatments we had a control diet, which contained selenium and vitamin E at what we would normally consider to be the requirements. Okay, so these diets were not deficient in selenium. They weren't deficient in vitamin E either. They were fed at what we would consider to be requirements. And we had 4 different treatments. So the control plus heat, then the selenium plus vitamin E, which was the thermoneutral, and then we had the selenium plus vitamin E plus heat. So these are the treatments under thermoneutral conditions, and these are the treatments under heat stress. So the first thing you see is that even when the temperature's say 26 degrees overnight, there is almost a 40% increase in respiration rate in the pigs that are exposed to the high temperature. As we increase the temperature up to 35 degrees, you see a dramatic increase in respiration rate. As I said, some are up around 200 breaths per minute. And then once we drop the temperature down again, the respiration drops away. This is in the control animals, and you can see that in those that were fed the high levels of what we call super nutritional levels of vitamin E and selenium, we can see a reduction in the respiration rate. Okay, it's still increased. We haven't completely ameliorated the effect of heat stress, but it is reduced, and this is significant statistically. It's also significant metabolically, and we've seen this in a number of different species now: sheep, cattle, pigs, and chickens. And this is the same study, and what we see here is that in fact under thermoneutral conditions we fed these animals at 800 grams per day, or 0.8 kilograms per day. Those sheep that were exposed to heat stress fed the control diet. reduced their feed intake by 14%, but those that were receiving the selenium and the vitamin E maintained their feed intake at the control levels. So not only were they did they drop their respiration rate, they actually also maintained their feed intake, and so their productive performance. So we're seeing quite clear physiological effects as well as production effects. The next slide is these are data generated by Fan Liu, did a PhD with us, and this is quite a busy slide, but I'll step through it with you. This is actually in pigs that were exposed to either thermoneutral conditions or heat stress. And the pigs were fed either selenium at 0.2 parts per million or 70 international units. Again, this is what we will consider to be the requirements for pigs. This is what pigs are normally fed, these levels. We increased the level of selenium 5 times up to 1 part per million, and we increased the level of selenium over 10 times up, vitamin E 10 times up to about 200 international units. Now, what we have on x-axis here is 5 kilodalton dextrose permeability of the small intestine. Okay, so we took samples of the small intestine and we were able to measure the permeability of that small intestine to molecules, in this case of 5 kilodalton size. And what you see in those animals that were fed the control diet is that when they're exposed to heat, there's an increase in permeability. Okay, so this means that the gut has been damaged because it's become more permeable. Now, as we increase the levels of antioxidants, we can see a linear decrease in permeability. And so, in fact, by the time we get to 0.5 parts per million selenium and 100 international units of vitamin E, we're back. The heat-treated animals are actually back to where the controls were. So we've actually reversed the effects on the gut. So with these relatively high levels of selenium and vitamin E, we've been able to reduce respiration rate and rectal temperature. We've been able to maintain feed intake. And in this model here, we've been able to show that we can actually reduce those negative impacts on the gut. A great story, except that the supernutritional levels of selenium that we've been using of around 0.5 to 1 part per million, they are greater than the maximum allowed in many jurisdictions. We can actually feed this in Australia, but in the EU and in the UK, we can only have 0.3 parts per million in the diet. So we've spent the next few years looking for alternative forms of dietary antioxidants that can at least partially substitute for selenium. And I have a lot of interest in plant-derived antioxidants as suitable alternatives. And I'll just talk about one of those groups of compounds that we work with. This is a different model again. This is in chickens. And what we did was we fed 6 different levels of a polyphenol mix derived from sugarcane. And we have chickens that are exposed to thermoneutral conditions or those who were exposed to heat stress. And we look at rectal temperature, average rectal temperature as you go along the study. What you see is that rectal temperature increases in those animals under thermoneutral conditions. It increases as the animals get older. And there seems to be some protection with the polyphenols. But in particular, when you look at those animals that are exposed to heat stress, a dose-dependent reduction in rectal temperature at day 36, also at day 28. The heat stress really didn't kick in until about day 21. So you can see that those chickens that were fed 10 grams per kilogram of this polyphenol mix had, about a 0.6 to 0.7 degree decrease in rectal temperature, quite potent effects. And it's not just in the physiology. Well, okay, this is another example of physiology. Sorry, I got ahead of myself there. This is looking at respiration rate under thermonuclear conditions around 65 breaths per minute, not really affected by the dose of the polyphenols. The control animals up around 170 breaths per minute, and then a linear decrease as we increase the dose of polyphenols. So quite clearly indicating that we're reducing rectal temperature and reducing respiration rate with this dietary treatment. And interestingly, one of the things that you see in animals that have been exposed to heat stress, that is sometimes the meat and muscle from those animals is affected as well. And what we're measuring here is T-bars, and that is a measure of oxidative damage. So that would mean that the shelf life of this chicken meat would be compromised because we get an increase in T-bars in the control animals exposed to heat stress, but we get a reduction as we feed higher levels of the polyphenols. So again, some effects not just on physiology. Right, and I'm sort of heading towards the end here, but just want to, I talked about diabetes before right at the start. So this is looking at, this is in humans. This is normal heat loss, or the heat loss that you see in type 1 diabetics as you increase the amount of energy. So this is exercise-induced heat loads. As heat load is increased through exercise, normal individuals are able to get rid of heat. They're able to dissipate heat, and you see that there is, a small increase in rectal temperature, but it's not too bad. If you look at the diabetics, though, they cannot increase the amount of heat they dissipate, and the primary reason for that is that they have very poor blood flow to the skin, and as a consequence, they accumulate heat and at high risk for heat stroke. So we've looked at using chromium under these conditions because during heat stress, animals may decrease their level of insulin secretion. And we know that improved insulin sensitivity is an adaptive response to heat stress through increased peripheral blood flow and also increased use of glucose instead of lipids as a fuel. We know that the chromium acts as an insulin mimic, and our hypothesis is that if we actually feed chromium, we can improve insulin sensitivity, increase blood flow to the skin, and help lose heat during heat stress. And again, this is work done by Fan Lu. Here he's got a control diet plus chromium, a control diet at 35 degrees, and a control 35 degrees plus chromium diet. And you can see that rectal temperature is decreased at the height of heat exposure, as is respiration rate. And this is quite a dramatic reduction in respiration rate. And we've done a number of other studies where we've shown some of these effects. So just to conclude, heat stress will increase in severity and frequency, presenting problems for both humans and livestock. Heat stress can have severe impacts on gut integrity. I think I showed that quite clearly. And this can be partially reversed by antioxidants and possibly plant-derived compounds, other plant-derived compounds. Heat stress increases respiration, rectal temperature. And again, these effects can be at least partially reversed by antioxidants and other plant-derived compounds. And chromium may also improve blood flow to the skin and reduce the physiological load during heat stress. So thank you for a whistle-stop tour through some of the work that we're doing in this area. I'm quite happy for you to contact me if you want to get any more information. And also, I think now that there'd be a short opportunity for a question and answer session.
Excellent. Thank you very much, Frank. So just to remind our audience, questions, please use the chat function to ask your questions, and then Frank will answer them that way. So while you're can't see any questions at present. So while we're waiting for questions to come in, I have a question relating to the data, Frank, that you showed on feed intake
Yep.
and oxidative damage to the mucosa of the ileum, I think it was.
Yeah.
That data implies that it's the oxidative damage to the mucosa that is responsible for the reduced feed intake, as opposed to the animal trying to reduce the heat stress associated with presumably heat increment.
I think they're both occurring, and in fact, some of the damage to the gut may be a result of from not eating. Because I know that the group at Iowa State often do pair feeding studies when they're doing the heat stress effects, and you will get a reduction in villi height just through removing food for a short period of time.
Okay.
But the effects of heat stress are greater than just the removal, what you see when you remove feed.
Right. Okay.
Yeah.
Thank you. Okay, we have a question. What is the form of chromium in the supplement, presumably chromium 3 picolinate complex?
It's chromium picolinate. We've actually we've worked with chromium picolinate, but we've also worked with nanochromium picolinate as well. Because one of the problems with chromium picolinate is it's actually it's quite difficult to mix in the feed because some of the particles are quite large. So we've made a nano and micro particle complex, and that's actually much more efficacious.
Okay, excellent. Thank you, Frank. And another question. I never know whether I'm allowed to read out the names of the people answering the question. So that question was from Steve Banwart, and we have one from Fiona Smith. What is the UK and EU's objection to feeding cattle more selenium? Are they concerned about animal welfare? And will these objections be removed with the shift to feeding plant-derived phenols instead?
Okay, so the worry about selenium is related to the fact that selenium in some forms is toxic. Inorganic selenium such as selenide and selenate, which is sometimes used as a supplement, can be toxic at the levels that we're talking about. We've been working with chelated selenium, protein-bound selenium, often in the form of yeast. I've also grown mushrooms on high selenium compost and got similar levels of selenium, and that's very safe. However, it's just the regulatory environment is that the toxicity does occur, and so it's a safety issue because I suppose you can't guarantee that the levels of the sort of selenium that people are going to be including in the diet.
So just to clarify, is that then in Australia you have have less stringent thresholds for the amount of selenium then?
Well, in Australia, most of the country is selenium deficient, and so historically, what we've had is selenium deficiency in our livestock. There's a part of Queensland where there's actually quite high levels of selenium in the soil, and so the toxicities occurred there, but they're just not the same sort of concerns, because it's just not been an issue we've seen. Yeah.
Okay. Right. Question from Christine Bosch. Heat stress duration. Are there short-term compensation mechanisms?
Yep. Yes, there are, Christine. You see most of the effects within the first 3 days. So if you look at the effects on the gut, they're dramatic and rapid. But after about a week, the gut does adapt. There still will be compromised performance, but they will adapt. In fact, one of the big problems in Northern Australia, where the temperature can get quite hot, we often see the problems in beef cattle, for example, when they come back on feed. So when the heat stress is finished, they go back on the full feed, and their gut is just not ready for that amount of food, and we get some gut damage then. I did see Stephen had a question about the chromium, and he's right. Chromium 5 is toxic. We're not talking about chromium 5 here.
Okay. All right. I've got a quick question, Frank. In relation to the way in which you administer these antioxidants, does it make a difference if you? I mean, all of the methods, I think, or certainly ones relating to a feed intake, you've added the antioxidants in the diet as opposed to say administering them directly into the circulation. Does it make a difference? I mean, I know you've said obviously blood flow moves away from the gut to the periphery during heat stress, and presumably therefore there are fewer antioxidants being delivered. Therefore, so does it make a difference?
I suppose the dietary route is the logical way, because some of the effects actually occur in the gut as well. But there has been, you can inject vitamin E, and I know that some researchers in the Middle East have worked with injections of selenium as well. But the thing about having in the diet is you actually get a more sustained delivery as long as the animals are eating. Yeah.
You still get the same benefit, irrespective of route of administration.
There's only really been a couple of studies done, Henry. So I'm probably not ready to say that yet.
Okay.
But obviously our interest in plant polyphenols is that if that makes an ideal delivery mechanism as well.
Yeah, I mean,
I
guess one of the questions is obviously with the polyphenols they're not their bioavailability is sometimes questioned, and as you said, if their function is actually in the lumen of the gut as opposed to within the mucosa, then it makes sense.
Because we saw those effects on meat oxidation, it indicates to me that at least some of the compounds are being absorbed.
Yeah. Okay. Excellent. Right. It is 12:40 UK time, which means I should wrap up this seminar, and there aren't any more questions to deal with. Lots of people thanking you, Frank. So just remains for me to thank you, Frank, very much for your seminar, and to thank our audience for listening and asking questions. And until next time, goodbye to you all, and thank you again.
Okay, thank you, everyone.